Welcome back, everybody. Fabulous to have you here. I know lunch seemed to go really quickly. Day seems to be going pretty quickly. But it's my tremendous pleasure to introduce Chirag Patel and Ashwini Davison, who are going to be moderating the session. I know from a number of people that I spoke to before the conference that these two people are both tour de forces, and so I think this is going to be a really fabulous panel.
Thank you so much. Chirag and I are really thrilled to have this panel here today. Our virtual participants who are taking time out of their busy schedules, as well as those in person. Again, I'm Ashwini Davison. My current role is as the CMIO focused on oncology at Amazon Web Services. I'm also part-time faculty at Johns Hopkins University School of Medicine and School of Public Health. Started at Hopkins in 2007. So thrilled to be here, and I just wanted to say that for all of you in this room, including the folks from foundations who have come here because they feel that for their mission-driven use case, this could be the home. This could be the home that perhaps pushes things forward. I see we have our interim president of the Pediatric Brain Tumor Foundation and others in the room who are strong advocates for incorporating environmental health, other factors, this larger exposome into the common vernacular, which it's not there yet, right? I liked the comment this morning, PCR during COVID became a part of the vernacular, and after this, we're gonna get exposomics beyond Gary and Chirag and Fenna and that cohort to really be a part of the vernacular, and foundations are gonna be a big part of that. So, I just wanted to say, Chirag, right, the purpose of this panel and our esteemed guests is to talk about the role of AI, big data. You prefaced it this morning, but I know you also have some thoughts before we throw it to the panel.
Let's do it. It's not hot.
Let's maybe do it. I mean,
Just start talking. All right, it's detection. Okay, great. So thank you, Ashwini. Thank you for co-moderating this amazing panel of our august panelists. So as you all know, earlier today I talked about this elephant in the room, which could work to our mutual benefit: artificial intelligence. It's clear it's come to make an impact in biosciences, biomedical enterprise writ large. With the advances of things like large language models, foundation models for language, and now things like AlphaFold, which promise to help make more efficient the scientific processes, for example, of learning how proteins fold, interact with other molecules. Seems like magic, but behind that magic was a lot of hard work of putting together benchmarks and data such that this program was AI ready. So the broader question for our panelists was what is it going to take for exposomic scientists and science enterprise writ large to make our work AI ready? We've asked our panelists to also opine upon the critical use cases of the field of data science and application to help answer this question about how to make things AI ready, how to reproducibly and efficiently discover the elusive environmental determinants in disease, but also think about lower hanging fruits of integration across different modalities, time, and disease models, as we discussed earlier. So Ashwini, I leave it to you.
great, thanks so much, Chirag. And for those that are just joining, welcome. We're not gonna address everything, believe it or not. We're not gonna solve the problems in this 45 minutes. That's what the breakouts are for. But really, this is an important discussion, and I'm gonna start with Murthy, professor of cardiology from the University of Michigan. And we were talking about this a little bit earlier. I said I'm gonna start with the triggering question, because it's important, because there's folks here who aren't on the clinical medicine side or the clinical decision support side, but we all have this end goal, right? This end vision, be it with Indipharm or others, to drive clinical decision support, be it with AI or not, with factoring in exposomics, with near real-time data. So Venk, with that eventual goal, how far out do you really think we are from seeing the exposome in its entirety being factored into driving clinical decision support at the point of care?
Yeah, that's a really great question. So I wear a few different hats, and when I think about each of those different hats, I have a little bit of a different perspective. So I'm a clinical cardiologist. I see patients with cardiovascular disease. I'm an editor at one of the journals in the AI space, NEJM AI. And then, of course, I'm a physician investigator as well, and I do research. And so there's a part of me that wants to say that this is going to, be there tomorrow. But I would actually divide the applications into two broad areas, right? There's the question of mechanism and discovery, where there's no doubt that the AI methodologies are going to have huge impacts. They're going to give us insights that we weren't able to get otherwise. They're going to accelerate things that were perhaps too laborious or time consuming to do otherwise. But when we think about the clinical problem, I think there's a number of hurdles before you get to the point where you say, we'll use this data in routine clinical practice. I mean, when you think about other types of omic data, for example, genetics, Dr. Wojcik gave a great talk this morning about how in some diseases this has made a huge impact. But in many chronic diseases, when we think about some of the priorities of the administration right now, but also just in a public health sense, from my cardiologist hat, thinking about obesity, diabetes, cardiovascular disease, hypertension, lipid disorders, these sorts of things, the vast majority of those, there probably are important exposomic variables. But the question is, how large are the effect sizes for these, and how do we integrate them with all those things that we already know? And I think that is a problem that remains unclear what the answer will be. It is possible that there are probably subsets of populations where the effect sizes are large, and that's where maybe guiding treatment could have a big impact. But I'm guessing that overall, on a population basis, for many diseases, it'll be small. But then there's this whole other realm of the regulatory barriers and the validation, right? So this is the editor hat I wear, right? Where you say, well, you look at FDA has now cleared maybe about 1,000 AI products with some form of regulatory approval for medical use. It's increasing every day, but many of these actually have not gotten traction in the clinical sphere, in large part because the data behind them may not be great, but there's also issues around reimbursement. It's extremely difficult to integrate these into many clinical environments. It's a huge educational problem. So to summarize, like, what do I think the prediction is? To be honest with you, I think we're going to see much sooner implications on public health and policy than we are going to see it used at the bedside in the clinic. I'm guessing that will take a decade or more to get to seeing impact, and it may not be as broad as one would dream of. There may be niches where there are large impacts, but in many cases it may be small.
Yeah, no, thanks so much, Venk. And, some of that is inspiring. Some of it might be daunting, right? But it's important for us to keep it real. And I think moving further upstream, I'm going to throw it now to Nicole, who's joining us virtually. Nicole, thanks so much as Acting NIH Deputy Director for Program Coordination, Planning, and Strategic Initiatives. Your work, even prior to this role, has focused on advancing predictive models that combine in vitro, in silico, and high-throughput screening. So the question Chirag and I really wanted you to weigh in on is, as we move towards defining and measuring the exposome, how do you see interoperability and scalability playing out across these domains, from model-based systems to human-based data? So would love your brief thoughts on that, and then we'll have time at the end for the panel to discuss further after we go through everyone.
Part of the answer is in the question that you just asked, in terms of emphasizing the necessity for interoperability at scale, right? And so I think that AI holds a lot of promise to integrate complex exposure data. So, we're going to be having data streams coming in from wearable sensors, from satellite imaging, from metabolomics, from electronic health records. And so building more advanced machine learning algorithms that can actually synthesize both structured and unstructured data across time and scales, like ranging from molecular scale to population level scale, will actually give us, the more multi-scale integrated, larger data sets that we can then apply AI from the perspective of predictive modeling to detect those patterns in those sort of noisy high dimensional data sets and uncover those links between environmental exposures and health outcomes. So I think that there's a role for AI and machine learning in sort of every step of that pipeline, from harvesting the data to curating the data to making it interoperable and scalable, and then doing sort of discovery, predictive modeling to uncover those sort of how health risks are related to your cumulative exposure trajectories, and then translating that into actual like precision prevention strategies.
Yeah, no, that's great, Nicole. Thank you so much. And I think it's a perfect segue to Marc Chadeau next. So across the pond from Imperial College London, Marc, you've really been working at that intersection that Nicole was just talking about, right? With pop health data science and cutting edge, exposomics work. Can you talk a little bit about what needs to be done with, environmental exposures in biology to actually allow for cohort-based data to be evaluated? To make this AI ready, but maybe AI also has a role to play, like Nicole was saying.
Yeah, thank you very much. Indeed, we've been working into many Exposome projects into trying to make the analytical tools ready for answering some of the Exposome research question, and we identify clearly. So we're a bit at a crossroad where we can make a bit of a look back what we've done and what's left to be done. And I think in terms of what's been done is identifying key gaps in the tools that we needed to have to do exposome profiling, and these related more to feature selection identified from large set of data, features or variables that are actually influential, either in understanding the response to a certain exposure or as a predictor for future chronic conditions. So this work has been done on this aspect. Also, as mentioned before, multi-dimensional, multi-layered type of data integration. So there's been a lot of effort done in that respect, and some algorithms are emerging as good ones, and also trying to reduce a bit the complexity of the questions of the data and get a kind of a simpler representation of the data, of the questions, to get more interpretable results from these inferences. So I think this has been achieved. It's been a shift in the methodological framework used. Now, with the emergence of far more data in terms of dimensionality with real-time monitoring, either of exposures or health outcomes, there is a need for further development of the toolkit that we have that can accommodate that longitudinality, if you wish. There's also an important role to switch from like more snapshot modeling to more of a dynamic approach. And finally, to try and make the, and this is where I think AI has a big role to play. And finally, when we want to put the exposome science a bit closer to the policy making, we want to have robust causal inference models and robust counterfactual models to try and estimate what could be the potential effect of a certain intervention. And this is where Gen AI as well could play a good role. So just to summarize, when Chirag asked me how can we use AI in exposome sciences, the first word that came to mind was carefully. So we need to be careful about what we do and why we do it. And that comes with training, with growing the culture and so on.
Yeah, no, great. Thanks, Marc, Nicole, and Venk. And I'm going to pass it off to Chirag.
Yeah, that was fantastic. I think there's a tremendous promise of using these new technologies as mediators or readouts of exposure to understand biology simultaneously. And I think this speaks to some of the gaps that, John, you might be experiencing. So John Wambaugh from the EPA, your work has been on identifying in a high throughput manner exposures connected with phenotypic readouts in vitro. And question for you, inspired by your work on ExpoCast, which is this platform, what are the approaches to you seem most promising, and where do you think the field needs to go with exposomics in hand to help us develop some of those benchmarks to take your work to the next level?
Yeah, that's a great question. I'm hoping that you can hear me. So with the ExpoCast project, we've been trying to predict what sorts of chemicals people get exposed to. There's literally thousands of chemicals in commerce, but we don't get exposed to all of them, but we do get exposed to some. We've taken a statistical approach of just looking what is the typical chemical that you get exposed to look like and how is that different from one that you don't get exposed to. And you can see patterns. When you look at the Centers for Disease Control data, for example, you can see that chemicals that are in our homes, that are close to us, that have sources that are near us, tend to be several hundred times higher than more chemicals that we might think of an environmental chemical that's released into the environment through a smokestack or something. And so we see those patterns even when we do non-targeted analysis and we look in blood. We might not be able to immediately identify all of the chemicals that are present in our blood, but we can see in some cases patterns that they are very similar to the sorts of chemicals that we see in consumer products. And so this is the point right now where Professor Murthy mentioned validation and Professor Chadeau mentioned being careful. If I was going to show one figure, it's this great picture of a piece of bacon that looks exactly like Elvis Presley. And the reason I would show that picture is that our brains are terrific at seeing patterns, whether or not they're there. And machine learning is very similar to our brains in that I could train a machine learning model to sort through pieces of bacon and find the one that looked most like Elvis. But at the end of the day, we need to be careful. We need to validate that that's really a thing. Does bacon really look like Elvis? Is that a meaningful thing? And so where I think we can deal with that is really in some sort of mechanistic insight. So it's, the pattern that we see is a hypothesis, but we need to follow that up. We need to talk to, in my case, I talk to exposure scientists and say, well, can you really get exposed to all of these chemicals in the home? And what you see, we've been able to build things like machine learning models for the functional use of a chemical. You give me a chemical and I can look at it and we can say, oh, that's a plasticizer, or that chemical is a surfactant, it's a soap, it's gonna make it good for washing things. So yes, it makes sense that we get exposed to it. And so by building models that have some degree of mechanistic insight when we're building our pattern classifiers, we can hopefully get some of that carefulness in there. And we're going to need machine learning to sort through the amount of data and to fill in the gaps we have, but we have to have other clever people to kick the tires on it and make sure we're not tricking ourselves.
Got it. Thank you, John. That was fantastic. I think that's a great segue to other new tools that the exposomics community are using to measure. One of them that has tremendous promise are high-throughput mass spectrometry, which, Michael, you've been working pretty hard on, in particular this area of annotation of things that we don't know about, or the unannotated metabolome. Can you speak to how you're going about this, and what tools can also allow you to scale up this search, at least for gold standard label data to help you feed into those models that John and others were talking about today?
Yeah, another great question. I think it's useful to think about what models we would like to have, and that in turn, will help sort of think through what data do we need to train those models. I think in terms of identifying small molecule exposures with mass spectrometry, really the dream would be to be able to identify previously unknown small molecules that are not in any existing database of known exposures. And I think that there's an increasing recognition that this is a major challenge. I would add that for these models to really be as practically useful as possible, we need to get to a point where they can return some well-calibrated estimate of the false discovery rate for each of these predictions. So what data do we need to make that happen? I think there is a recognition in the community already, and there are several large-scale initiatives to assemble more annotated tandem mass spectra for known small molecules. I think what's less appreciated and what's perhaps a big opportunity is to create label data sets that have known exposures in the context of a full metabolomic experiment, and that would provide the sort of data that one needs to leverage additional sources of information that are captured by mass spectrometry, but which are really treated in a very ad hoc manner, or perhaps not even used at all by sort of state-of-the-art models like retention times, MS1 information, and that sort of thing. And then I guess finally, a big bottleneck is going to be confirming these predictions, and that's going to require close collaboration between, computational scientists and experimentalists.
Fantastic. Thank you so much, Michael. We're going to switch gears a little bit. So in our panel, we aim for breadth in a bit of depth. So switching gears here, ultimately, or some of our experiments might need to be done in what are now silos. And so we have here Prashant Shah from Intel. And in general, a issue in health AI and implementation, or in both in implementation and discovery, is inferring over multiple sources of siloed data. So Prashant, can you take us through how you might envision these new collaborative models occurring in a federated and secure way across these what are now potentially silos, but unlocked across locations to increase power for detection?
Yeah, thanks. Can you hear me okay? Okay.
Yep.
Data for exposomics is, of course, multifactorial, right? And a lot of the factors are private. Genomic data, clinical data, all of these data sets are private and are governed by the local regulations of a given country, of a given institution. And so moving all of this data to a central location is generally very challenging. And of course, exposomic data is very dynamic. So moving all of this data around can be fairly challenging. And for us to be able to actually create robust AI models or analytics, you need access to large amounts of data. And it's not just a large amounts of data, but also the diversity of data, because you could have very different sort of exposures in different parts of the world, and you could have different socioeconomic context. So how do we increase the diversity of data that we can make available for these type of analytics or even for machine learning models? And centralization is, of course, one approach, but it's very hard to centralize. So federated learning essentially is a new paradigm by which you are letting the data stay where they are, and you are instead of bringing all the data in one location and then analyzing all these data sets, you're instead keeping the data at its source, and you're sending the machine learning model or the analytic query to each of the data silos, and then performing some sort of a joint analysis based on the sort of results of that. So this can essentially increase the generalizability of these AI models. It can increase the robustness of these models. You can even validate these models on private data sets around the world to ensure that they're not just working very well in one local context, but is actually working well across multiple different countries or regions around the world. So, and then one more place that is really promising is that it can really, like, sometimes you have fragmented data sets around the world that are, let's say rare exposures, where you don't have a lot of data at one given site, but in aggregate around the world, you may have, small data silos. So you have a small data problem. And so by using federated learning, you can essentially combine these rare exposure data sets in a manner that preserves privacy and yet allows you to actually unlock science, right? So one such study that we've been involved in is essentially the effects of space radiation on astronaut health, and what are some of the genomic genes that are basically implicated in astronauts developing cancer due to space radiation. And there's no one site that has a lot of astronaut health data. And so, European Space Agency has some data, Mayo Clinic has some data, NASA has some data, but these are very small data sets. And so by doing federated analytics, we've figured out 2 new genes that are implicated in cancer, in astronauts developing cancer due to space radiation. And so then NASA actually shipped a server with federated learning software on the International Space Station, because they are also acquiring a lot of biological data in space. And so that's the type of use case that federated analytics can essentially enable, which is promising for the field of exposomics.
Yeah, no, that's great. Thanks so much, Prashant, Michael, and John. I think that was a great way to segue to, one of the efforts that I'm involved in is with the Cancer AI Alliance. So another one where federated, not centralized data modeling is the approach. And so this is, one of the areas where I'm most interested in, because I think to Venk's point earlier, right? Like, are you going to have the volumes that you need, and how can we, share, AI insights across cancer centers without sharing the underlying patient data, because that is sensitive data. And so it's not just the issue of, transferring the data, but the security, the redundant copies. And so with Cancer AI Alliance, it includes Fred Hutch, Memorial Sloan Kettering, Dana-Farber Cancer Institute, and Johns Hopkins Medicine. And so I'm hopeful that, you have that space use case and truthfully, moonshot oriented. And one thing Gary and I have always talked about is the need to collaborate with those that are already doing something, right? And so where is that interest of these collaborations where we're trying this federated learning approach across NCI cancer centers, and as they expand from just EHR data, MOB common data model to including imaging, genomics, can we get, exposomics in there? Are there, is there motivation for those use cases? Piloting it with 4 sites, but eventually, right, the goal is to go to all 50 plus NCI cancer centers, the 1,000 additional, cancer centers in the US, and then globally. So I really liked what you said, Prashant. I think this concept of federation is so important. And I will say, Chirag, this morning you talked about the need to move upstream, not only in population health, but in medical education. And I think this is a concept that, is not something that everyone's familiar with. Secure computing, how do you do differential privacy, what are those capabilities? So really great kickoff. And in that vein, I really wanted to support this panel. And Chirag, you're gonna be okay? Because I have to go to a Cancer AI Alliance call that's actually, to unblock something that's related to the secure computing concept. So this is near real time, and we planned it this way. You're gonna be okay?
I think I have a great group of people to help me out here, but
Yeah,
you got to
okay.
do what you got to do.
Yeah, no, and I will see everyone at the breakout today, this afternoon. I'll be at the AI one, and look forward to continuing the
Sounds-
conversation.
Yeah, absolutely. Sounds fantastic. Absolutely. So one area that we talked about, or at least touched upon, is the use of AI for precision or getting better, for lack of a better word, health outcome and phenotypic information, phenotype data. You spoke to this, Marc. I know on the other side of the research program is getting a better sense of predictors for exposure. So I know, Venk, you have predictors that you didn't talk about today that link, questionnaire items to exposure data, which is going to be impressive to help us understand potential biological linkages to different outcomes by using that as new predictors. To that end, Tim Althoff, who couldn't join us today, he's been doing a lot of work in use of AI for phenotyping, and he mentions today he couldn't make it due to an emergency, but he wanted to share how generative AI specifically provides an opportunity to also engage with consumers about their phenotypes. So to actively collect non-traditional sources of information that can be helpful in understanding of psychology that was developed through use of a large language model, and that he's deploying in practice today. So I thought that was a particularly exciting use of better phenotyping, as well as getting to better indicators of exposure through AI tools. So with that, let's open up to the panel. Did you what resonated throughout the panel with what all of you were saying, and I'm sure you have tons of questions for one another. Let's hear them out. Let's start with Venk. Could you tell us about connecting the internal and external exposome and some of the work you've done there?
Yeah, I mean, I think this is incredible opportunity that we have the ability, as many on this panel and in the audience have pioneered the ability to make measurements of thousands of, or hundreds of to thousands of molecules in the internal exposome. And I think when you think about how we do research more broadly, one of the hard problems in cardiometabolic disease is diet, right? You can do what our ability to do research studies on diet and nutrition is always challenging for a number of reasons. There's recall issues, there's adherence issues. You know, I was talking to some folks about how would you prove you know, that one diet was better than another approach. And traditionally we've done, you know, maybe feeding studies where you provide meals for a short term, but that only gives us surrogate markers, right? And if you try to do this longer term, people drop off on their adherence. But by using these kinds of markers, we develop signatures of different dietary components in some cohort-based work we did, and that allowed us to identify markers that potentially were surrogate markers that were more closely associated with clinically relevant phenotypes, and take those to other cohorts for validation, and potentially in the future use those as markers both for what somebody is actually eating as opposed to what they're telling us they're eating, but also in prospective studies to assess whether adherence is modifying the benefit of a particular dietary approach, for example. So I think these kinds of things of tying together traditional questionnaire-based ways of assessing the exposome and more novel kind of internal exposome measures, actually they're synergistic, they're not necessarily competitive.
Absolutely. Any questions from online? I didn't. All right.
I just
John,
wanted to
please. Yes.
Yeah, I just wanted to offer one thing that as we look for patterns, especially as we get new data, one thing, and I'm very optimistic about new data sources, but part of the patterns we will see will be associated with how we make the measurement. And so, you know, one of the first things we'll see is, oh, I can see that this was gas chromatography versus liquid, right? You know, those are whole different chemical spaces. And unfortunately, we'll be able to see, oh, this was technician who comes in on Mondays and Wednesdays, and this was measurement was made by the technician on Tuesdays and Thursdays. And that's okay. The fact that those signals are in there is okay. They're confounders, and we just have to be open when we're looking for the patterns that we initially want to find, the patterns that are just, how did we come about making that measurement? And once we subtract that background off, that's when we begin to see, oh, this was dietary intervention number 1 versus dietary intervention number 2 versus a third person who said they were doing the diet and never actually did. And so approaching the problem, knowing that we're going to find the confounders is I think just, and I know the panel knows this, I just wanted to state that's eyes wide open.
Oh, no, absolutely, John. Get to you in one moment, Nicole. This prompts a question for the broader panel about, and maybe we can begin with you, Marc, about like in this new era of AI, it's like, you know, there's this debate, often useless, about, oh, AI is going to solve all these issues. But what it comes down to ultimately will be about study design. Mm-hmm. And so you talked a bit about, like, you know, these new models you're developing of tracking people dynamically, mm-hmm. you know, through the life course potentially. What are your sense of the study designs that are out there that could help us scaffold these new AI implementations?
So, well, in the study design for sure, prospective cohorts with linkage is really all these big power banks where you have a lot of data at baseline and a very detailed follow-up of the health of the individuals is a precious material to run this kind of analysis at a large scale. What I think is very important to be done, and we're just starting there, we're generating huge amount of data with a resolution that may be a bit unreasonable in the sense that maybe we don't need that. So I think what we need to ask ourselves, and this is where definitely pattern detection algorithms could be very, very useful, is to identify for a wide range of health outcomes, what are the key features in your high resolution exposure profiles that are actually predictive and predicting more than usual summaries, cumulative exposure kind of things. So this is where the first application could already be done on large data set where you have that linkage done. And then the second real good application, I think, is really about counterfactual modeling and try to quantify the effect of an intervention. And Gen AI is really about mimicking the data generation process. So you could definitely use that to well, that's the whole digital twin stuff, but basically use that kind of models to plug in and out some exposures or some interventions and get that's an easy win, I think.
So instead of creating your own images, you're creating your own data sets? Is that what you're doing?
Yeah.
Cool. Nicole, sorry, go for it.
No, this is actually a perfect segue. It's very relevant to what I was going to say, which is that when we're looking at study design, obviously, I think there's a lot of opportunity and potential in looking at things like population level interventions and tracking sort of the signals that the predictive models are giving us and whether or not they're borne out in human populations. But I think something that we cannot forget is the necessity of interfacing with experimentalists, particularly in the burgeoning field of human biology-based alternatives to animal models. So when you look at where I think we're going to see a lot of success in testing out some of the hypotheses that are generated by AI and machine learning when we apply machine learning models to these large multifaceted data sets, we're going to get signals that say, oh, we think that these chemical exposures are potentially associated with, for example, these neurodevelopmental disorders. And now, increasingly, with microphysiological systems and things like brain organoids, we have the ability to actually experimentally query those associations that come out of those predictive models in human biology-based experimental systems. And that's not specific just to the brain. That was an example. I mean, we have lots of microphysiological systems that target various organ systems in the body and the ways that they interact with each other. And having that paired with computational systems models and digital twins in a way that is iterative and cyclical and allows for both hypothesis generation and hypothesis testing and refinement is, I think, absolutely essential to the success of this effort.
Awesome. Those are way out there ideas, but I think appropriate for this type of program. Let's open it up to the audience, and before that, I would like to pose a question too to the panel, pointedly to Michael, and I think we can all have our opinions on this. Is that the panel is prolific. All of you guys have been publishing fantastic papers in great journals, and sort of have an eye on the prize of like how what is the potential. But what is the burden of proof ultimately in your take? So you talked a little bit about this, Michael, about like false discovery rates and things like this. What are the measures you think that are ultimately going to be that we should be looking forward to as we push towards these new high throughput measurements of exposures? What should we be looking at?
Yeah, another great question. I think my answer is going to be a little narrowly focused on a very broad question. You know, again, thinking specifically in terms of mass spectrometry. I guess you can differentiate between showing that your model generally tends to make good predictions, so things like estimates of the false discovery rate or estimates of its calibration, and then the ultimate experimental validation of those predictions. And the latter, of course, has to really take place through a collaboration with experimentalists, and has been a real bottleneck in this general area of identifying these unidentified peaks that I think many in the audience are probably familiar with.
Yep, absolutely. All right, questions so far? Great. Go for it.
Thank you, Heidi Hansen from Oak Ridge National Laboratory. I really appreciate the comments that you all have discussed. I do have a question for you. So there's the promise of AI and digital twins, and then there's the challenge of AI and digital twins and really building something that can move the science forward. I think those challenges are surmountable within a 5 to 10 year time scale. But if we want this to succeed, we need short-term wins. So I'm going to say, can you give me an example of something that I could use AI with now that I would be able to have high fidelity data, a large volume of data that I need for my AI model, and that I would be able to get that short-term win to show folks that this is really something that's very promising?
It's a fantastic question.
I mean, I think there's really rapid adoption of AI in a number of spaces in the research arena, right? So identifying, when you think about real world data or even cohort-based data, identifying and quantifying exposures We can do that now in cases that were extraordinarily difficult to do before, and the same way with outcomes, right? So that isn't necessarily exposome. Those measurements are being made, but they will facilitate exposomic research in a major way. And I think that's happening today.
Can I add one more constraint? Something that we can validate. So I think a lot of the measures we talk about and exposures we talk about now, they're hypothesis generation. But if we're building something it's hard for us to explain what's going on, how do we pick or optimize what we want to go after in terms of testing those hypotheses? And if we want to do that, we have to be able to validate and be able to say we really think that what the AI is generating is trustworthy.
Maybe it's not exactly an answer to you, but I'm quite skeptical and careful about AI. I just not like want to do it, whatever it costs. But I think there are some successes and some easy tasks it's very good at. For instance, evidence synthesis, or can use powerful LLM models to get a good summary of a corpus of papers that would take a few lives to read and understand. So these kind of tasks already are successfully done and validated through human checks of some of the analysis.
I would add to that areas of computer vision also.
Absolutely.
I also have some very nice applications. Over on this side here, yeah.
Yeah, Kirsten Overdahl from NIEHS. So I am a mass spectrometrist. I am not a creator of tools. I'm a user of tools, specifically in the space of de novo annotation for previously uncharacterized molecules. And so my question is kind of thinking a little bit more about the collaboration between creators of these AI tools and then experimentalists, specifically kind of thinking of the old phrase garbage in, garbage out, and thinking about the outer applicability domain of some of these tools. And I'm thinking specifically of recent examples I've had in my own work where I'm using multiple tools to try and annotate a chemical completely de novo, no previous annotation in a fully untargeted data set. But ultimately the tandem mass spec data wasn't good enough in the first pass. And so we needed at least some clue of what class of molecules we might be dealing with in order to then go back, refine the analytical assay, try to get better tandem mass spec in order to do the de novo annotation, because what we saw was that multiple tools each gave us a different answer for what that molecule would look like, not even with specific moieties, but completely different classes. And so perhaps, Michael, the question's best directed to you, but what do you think that feedback loop looks like iteratively as experimentalists and tool creators come together to create tools that try to conquer some of that?
Yeah, that's a great point. I mean, I certainly agree with you that the point was made earlier that machine learning at its best can mimic how humans think, and we're not in a position to develop that sort of technology unless we understand how, expert analytical chemists really interact with the data. So I think it's a very important point that you raise. I don't know that there's a simple answer other than to bring computationalists and experimentalists together as much as possible and try to foster that exchange of perspectives and share how these two very different communities look at the same data in very different ways.
Awesome.
Are we done, or can I throw in one comment on that?
Go for it, John.
I have a colleague at EPA, Dr. Kristen Isaacs, who does what she calls exposure forensics. And in that case, multiple AI, well, where are you doing machine learning? But multiple machine learning models can sometimes help you. So if you have a list of small molecules and you're trying to figure out which ones they could be, and I mentioned functional use, which possible molecules might be there from a standpoint of, well, these would serve some sort of role. Well, we also have models for media occurrence. What sort of chemicals actually occur in, say, drinking water? And if you're looking at a drinking water sample, rule out the ones that only occur in air or only occur in some other media, so that you can start to use, if you build a suite of models that understand small parts of the universe, and then you can start to do the forensics where you allow the different lines of evidence. Okay, I measured this with a liquid chromatography. I measured it in drinking water, and it was near a factory that made hammers. You can start to understand some of these things. And so although any one machine learning model should definitely be looked at with skepticism, multiple lines of evidence can maybe be one way forward.
Absolutely. Thank you, John. So we're officially out of time, but if you could, if you have questions, if you could, for the record, describe your question, and we'll end the session. So did you guys have questions? Yeah, please.
Sure. My question is, thinking about the Gartner hype cycle, how do you communicate this without over-promising and under-delivering, especially as policymakers and average scientists start to pick up these tools?
Yeah, so shortly, my question is, should we actually forget the metadata of data and incorporate that as a part of the data? That's the very short, because that's the problem what we face. And it was, I think John was mentioning that already, that we detect the way how the measurement was done instead of the actual thing what we wanted to detect.
Yeah, and a question over
There's a
here.
question back there.
My question was how we incentivize and fund people using existing things, sometimes from other subjects, and making them applicable or better for either their own subject or the general model.
Got it, yeah. Oh, yeah, sorry.
Yeah, another question is in terms of trying to get structured data to begin with, or create practices for standard operating procedures, to what extent is there an appetite to try and break some silos with commercial partners or industrial partners who otherwise would keep it behind IP?
Thank you. Thank you all very much. It was great.